And so I want to welcome the first speaker, Doug. I got your name right. He's a rising senior at Jupiter High. And I think he was the great person for this project because he's really stubborn and he can make things work. He's not just only a great student, but he's also a volunteer for the Else for Autism. He's a camp counselor for karate, so I was like, okay, let's try not to make Doug mad. And I think he did a great job, so please talk to us about what you did this summer.
Good morning, everybody. My name is Douglas D'Amelio. I'm a rising senior at Jupiter High School, and I've been interested in neuroscience ever since I joined the Pre-Med Academy freshman year, particularly in prosthetics that detect nerve impulses and translate them to coordinate movement. Here at the Institute, I've had the opportunity to work with wonderful scientists and cutting-edge material. I've been able to talk to these scientists and researchers and figure out what they're doing and why they're doing it. Today, I'll be talking to you about a software called syGlass, which I will go more in depth later on in the presentation. It is a virtual reality software that allows for image analysis and research. Before I begin, I'd like to thank my mentors, Naomi Kamazawa and Kanan Thomas, as well as the rest of the EM Corps for providing such great images for us to analyze. And to my overseer, Ilaria Drago, I would like to say thank you for picking me to represent Max Planck this summer. I'd also like to thank all of Max Planck's supporters for making this program possible. And finally, I'd like to thank Max Planck as a whole for having such an amazing institute with incredible researchers that have been able to teach us and guide us throughout these past 6 weeks. It has been a once-in-a-lifetime experience and something I will value throughout my life. The experience and skills I have gained here will be crucial in my future career. The problem I worked to solve over the past 6 weeks is that of the inability to properly analyze data based on 2D images. Since data collected here at Max Planck and in research facilities around the world is in 2D, we lose a lot of important data when analyzing in 2D. Data presented on computer screens or on sheets, like the images we see here, are analyzed one at a time in order to see the full image. This in turn slows down the data analysis process and can lead to missing information or the missing of information within the study. The solution to this might be virtual reality. When most people think of virtual reality or VR, they think of video games. But the reality is that VR can be used for so much more. Since our world is in 3D, looking at images in 3D resembles that of our world. In VR, it not only lets us see this data, but manipulate it as well. I've worked with a specific VR analysis software called syGlass. And over these past 6 weeks, I've come to understand it far greater than I ever would have thought, and to some extent, may be considered an expert. Now I will talk about the software I've been using, syGlass. This software is a research and analysis software that can be used with or without a VR headset. Me and my partner Justin had a direct line of contact with the head developer and his lead engineer. As high school students, this was truly an incredible experience as we were able to develop and help them overcome a few of the bugs they were having. So how does it work? Data collected by imaging devices are 2D photos that are most commonly viewed on computer screens. Here we have what is called a Z-stack. While this term is only applicable to certain types of imaging, the concept is the same. Think of a building. Each image is a floor of the building, and alone, they are 2D and difficult to analyze. One would have to go through, say, 200 or more images to get the full picture. But when stacked on top of each other, they create a 3D image which can be analyzed with syGlass. Here is a quick example of how this works. This image is a GIF, and it is in 2D, and as you can see, color appears to be moving through the picture. You can see how the green fades away before the red does. This implies that the structure is on top of the red. And this image is of a nuclear pore, which I will go on later in my presentation. Having become well-versed in syGlass and how it operates, I was tasked with creating educational outreach videos for both the public and scientists to learn about the work we did here at the Institute. I created 4 videos while working here, one on basic brain anatomy, which me and Justin worked on, and 3 more videos on the different type of imaging we use at the Institute. I will explain these 3 different types of imaging and how I use them to further my work at the Institute. First, we have an MRI, or machines commonly found in medical and research facilities. They are used to scan either soft muscle tissue or the brain. The basics of how they work is by using electromagnetic waves to detect and record tissue. The first image we see on the left here is a 2D MRI scan, which was taken from top to bottom. As you can see, only one layer is visible in this image, but if multiple images are taken of different layers, it allows us to create a 3D image, which you will see on the next slide. The image on the right was cut from left to right as well. And this, combined with known data about the brain, allowed us to create the 3D reconstruction we see here at the bottom. And this manipulation allowed me and Justin to create our short educational video. As seen in this video, 3D manipulation is similar to that of real life, being able to hold, move, and change the size of whatever you are viewing. From the data in the MRI and known data, as said before, we were able to create a 3D model of the brain, which you will see at the end of the video. Here, we can see the cutting from left to right. And in just a second, 3D model will appear. And there it is, placed inside the MRI scan. It can be analyzed in real time. Next is fluorescent microscopes. Using fluorescent dyes, this type of microscope produces images by measuring the amount of light that is released by the dye when exposed to a specific wavelength of light. Here is the nuclear pore from the previous slide. One of my fellow interns, Alana Ferguson, has taken this image on the STED, a super resolution fluorescent microscope, which she will explain in greater detail in her presentation. The nucleus of the cell contains these pores that transport proteins in and out of the nucleus and the cytoplasm, the water-like substance within the cell. The red is the pore itself, expressed by a specific protein called nucleoporin, which is the fundamental protein responsible for building nuclear pores. The green around the pore is the Golgi apparatus, or the post office of the cell. It is responsible for packaging and transporting proteins and molecules within the cell. This video shows how we can observe the pore from multiple angles instead of just one. While a 3D model might be hard to construct with this, we are able to count individual protein within the cell itself, or the pore itself. And once again, this type of manipulation allows the data to be analyzed and visualized far greater than 2D. Finally, we have electron microscopes. By using electrons to see, we are able to achieve a resolution much higher than that of both MRIs and fluorescent microscopes, being able to see beyond a cell and even into structures within them, such as mitochondria. On the left, we have a transmission electron microscope, or TEM. And on the right, we have a scanning electron microscope, or SEM. Today, I will only be focusing on images from the SEM. A process known as serial block phase scanning is used to produce the 3D reconstruction of an otherwise 2D image. The image you see here is of the calyx of Held and the surrounding tissue. It is able to show in-depth parts about this structure and how it works. Being able to look inside the cells provides new opportunities to study and learn. The calyx of Held is an auditory synapse in mammals that transmits signals from the auditory system to the brain. In the video, you can see how the 3D image was stripped away to reveal our 3D model. Removing the 3D model from the 3D image, I then reconstructed the image to reveal blood vessels and neurons that are being constructed. This allows something that is impossible to see in 2D. Being able to trace blood vessels or neurons through multiple layers provides an opportunity that far outweighs or is far more beneficial than 2D analysis. So to conclude, I've had a wonderful time here at the Institute over the past 6 weeks and have learned that VR is an essential part of not only medical imaging, but research purposes as well. syGlass itself is an up-and-coming virtual reality software that is still under development and has been incredible to work with, despite some of their minor bugs. I believe that the future potential for this application far outweighs its current limitations. And syGlass, as of right now, is the only research software that allows for this 3D analysis. And we are one of 100 organizations that have access to this software. The potential for VR, and not just syGlass, extends far beyond research opportunities, but to schools, companies, and even to personal use. I will now take any questions and be happy to answer any you have later on.
While we see if someone has any question for Doug, I want to remind you that unfortunately for this project, it was very important for us to show you to wear the glasses and to see how it really work, what they did during the summer. Unfortunately, with the mask, we are not able to do it, but we invite you back for our next event because Doug and Justin needs to come back so they can show all the videos that they did during the summer. It is the big thing. So you need to be back. Any question for Doug? There is one in the back. Table out first. Yeah, thank you.
Was the video made while you were wearing the virtual reality? It had kind of that sense of I was still looking through your vision.
The question was if the video was made while I was looking through the VR headset, and yes, the software itself allows for what's called a narration to be made where it projects what you see into the video and your movements.
What was the most surprising thing you learned in this project?
The question was, what was the most surprising thing I learned during this project? And it was how much data can be analyzed at once with this. Instead of looking at, we had images from something that my partner Justin will talk about that contained over 350 individual images, and we were able to analyze all of those images as one 3D model in about 10 or 15 minutes.
Okay, thank you so much, Doug.